So now that we have covered bacteria, that leaves viruses as our other sort of pathogens. They're the other category that is left. So when talking about viruses, let's talk about some general characteristics. And in general, viruses are not considered living things, which is kind of hard to grasp sometimes, but instead we want to call them biological entities. And because they don't fit all the characteristics of living things. So if you recall, we had our song, the tune of Row, Row, Row Your Boat, where cells respond, grow, and reproduce, use energy, have DNA, adaptive living dudes. So viruses are not made of cells, so that alone is one of the reasons why they are not considered to be true living things, instead biological entities. So they do, however, contain some sort of genetic material, and it can be either in the form of DNA or RNA. Either one is fine. Both happen. And then they also depend on a host cell to survive. So that's another reason that they are not considered living things, because they cannot survive on their own. They require a host. And a virus is a virus that specifically infects bacteria is something that we call a bacteriophage. Bacteriophages are actually one of my favorite type because they look like little spider robot aliens, which sounds funny, but you'll see shortly that I'm not wrong. Spider robot aliens is very accurate for them. Now we can continue on talking about viruses in terms of their structures. So viruses in general take on many different shapes and sizes, but they all have these two things in common. So here we have a variety of different viruses, and this one over here is the bacteriophage that I said looks like a spider robot alien, which I think is very accurate. So depending on which type, they will still always have these two things in common. And that is that they will always be made of what is known as a capsid. And the capsid is the protein coat that is found on the outside of the bacteria. So you can kind of see on the different pictures here, they're in different spots, but they still all have a capsid. And then they also have a nucleic acid core, which is made of either DNA or RNA. So all viruses have these two things in common. When we talk about viruses, they still have many uses for humans. It's just not as many as when we're comparing it to our bacteria. But there are still ways in which humans use viruses for our own good. So to start off, scientists are conducting research to find ways to use viruses in the good of treatment of diseases, so things like cancer and genetic disorders. And they're also being researched as a potential replacement for antibiotics, which are used to treat bacterial diseases. So we are trying to harness the virus's own abilities to treat things like cancer. So if viruses can attack our cells in ways that we don't want to, can we make them attack cancer cells in ways that we do want them to? And the same thing going for antibiotics. Can we possibly use a bacteriophage to kill the bacteria that are in us without then harming us? So there are ways that humans and that we're using viruses for our own benefit. The last general idea that we need to talk about for viruses is viral reproduction. And it will always happen in the same sort of general process, and then we will also talk about how it can differ. So first step of this is the attachment phase. And in attachment, our capsid is going to bind to receptor proteins on a host cell. So our capsid is that little outside protein coat, and it's going to look for that on a host as well. And then step 2 is the inject phase, and this is where a virus is going to inject its DNA or RNA into a host cell. And then we can move on to step 3, which is to assemble. So viral genes are going to first be transcribed, translated, and then expressed. And from here, our host cell will become a virus-creating factory. So if we remember the terms of transcription And translation, that is what's taking your DNA and actually making it into proteins. So that's the step that's going on here in assembly. Then in our last step of step 4 is where our host cell is going to burst, which means it's going to kill it. And when it dies, it's going to release these newly formed viruses that it just made. As I said, this 4-step process is occurring in all viruses, but it can be slightly different depending which type you're talking about. So this is specifically describing a lytic life cycle of viruses, but there are 2 different life cycles which we will continue to talk about. So the first specific cell type we're going to talk about is the lytic cycle. And I shouldn't say cell right because viruses are not cells. So the first virus life cycle we're talking about is the lytic cycle. So lytic happens when viral DNA or RNA, either one, is injected immediately, starts being transcribed and translated. Okay, so the key word here, and it's in a different color, is immediately. So there's no lag time. They're just going to immediately start assembling and going through all of the different steps. So from here, our host cell, as we said, is going to burst, or lysis is the fancy word for it, but it's going to die, it's going to explode. And because of that, our virus cell is going to affect more cells. We were going to be releasing all of these cells, or all these other viruses out into the body, and that's going to go and affect more and more cells. The other life cycle that is an option is called the lysogenic cycle. And this happens when the viral DNA or RNA is injected and becomes a part of the host cell's DNA. So far, it's the same sort of process on the previous slide for the lytic cycle. But now, every time the host cell copies its DNA, it's also going to be copying the virus's DNA. This allows the virus to go dormant in your body until it is expressed. So what is different here is that it's stopping all of the other phases and it's just sitting in that phase where, so it's just copying DNA and copying DNA, but it never actually gets to the assembly portion where it's actually going to be sending cells out into the body. So it's staying dormant, and there are many examples we can see in our body of things that work like this. So chickenpox is a virus that we know you get as a child, but it can come up again later in life as shingles. Another example of this would be cold sores, where you always still have this virus inside you, but it's laying dormant until it has reproduced enough DNA and it's going to attack at once. So it's going to happen when you have a compromised immune system or stressed or something, and it knows that it has the ability to actually take over. So when it is lying dormant for a period of time is how we know it's the lysogenic cycle.